Well, hi everyone. This week we've been talking about cohort studies. And for the entire course, we've been using a data set based on one cohort study, the Framingham Heart Study. So you've become very familiar with that cohort, but I want to give you the opportunity to hear about another famous cohort from this area, namely the Nurses' Health Study. And with me is my friend and colleague, Mayor Stanford. We were students together, and he's going to tell you about the Nurses' Health Study. But first, I'm going to ask Mayor a couple of questions, and if he would share his opinion with us or answers with us. Mayor, I understood that in your former life, you were connected with the Framingham Heart Study. So you have the opportunity now to comment personally on the interactions of both studies, how they are implemented, how they are executed. And I'd like to hear what you think are the advantages and disadvantages of those two studies as you go through the description of the Nurses' Health Study. But more importantly, what made you want to become an epidemiologist? I mean, you are a physician. You could have had a career as a practicing clinician. What caused you to see the light, to join the other side and become an epidemiologist? So if you wouldn't mind sharing some personal perspective with us. So I'm going to leave and turn the floor over to Mayor to talk about himself a little bit, but more importantly, to talk about the Nurses' Health Study. So Mayor, it's all yours.
Well, to answer your second question, Fran, I don't see it as the other side. I see epidemiology as the primary care physician of the population of the world. And really, when that insight came to me was in medical school, I was really interested in prevention, but there didn't seem to be any good avenues to go where prevention would be the primary way to practice medicine. And it wasn't until my fourth year that I did an epidemiology elective when it dawned on me that epidemiology was really the way to practice prevention. So my whole career has been devoted to expanding the realm where prevention could be practiced. And really, this is what I've been trying to do my whole time. So yes, I was Framingham physician number 88. They're well now into their triple digits, but I'm an early pioneer two-digit guy. And I was privileged to work there and write some scientific papers with that group. The Framingham Heart Study has done enormous advances in epidemiology and our understanding of risk factors, not only for heart disease, but for other outcomes as well. The primary strength of that study is the close, intense follow-up of the cohort members over many years and even generations. However, it does have an important limitation, and that is the size. With an original size of a little over 5,000 people, that was enough to study common outcomes like blood pressure, high cholesterol, and even heart disease, which is the leading cause of death. But a cohort study that size really is not sufficient to study outcomes that are common but less common, such as breast cancer, cancer at any site, really. And it's also too small to get at some of the important subgroups. So for this reason, a different kind of cohort design was initiated back in the 1950s, which was the large, quote-unquote, simple cohort study. And this was the first big example was Sir Richard Dahl's study of British physicians, which was the first cohort study that established that smoking causes lung cancer. Now, Dahl was working on that study in the 1960s, and that was when oral contraceptives first came on the market, and he realized that this was a natural experiment in women where there was really no data whatsoever on the long-term effects of oral contraceptives. So he turned to his then fellow, Frank Spicer, and said, why don't you set up a study in women that would be comparable to the British physicians? So Frank thought, oh, that's a great idea. And he set up a study. Initially, it was going to be doctors' wives. So he sent letters to a large number of physicians, asked them to give questionnaires to their wives and have it sent back so they could become the cohort. And in the course of this pilot study, what he found was that instead of giving the questionnaires to the wives, the doctors were filling it out themselves. Helpfully, they thought. But in fact, it really invalidated a lot of the data because we really needed the responses from the women themselves. So that's when we turned to nurses, and the Nurses' Health Study was established. So I'm going to describe the concept and the advantages and the limitations of the Nurses' Health Study, show you a little bit about the operation of the study, and describe just a couple of the important findings to give you a flavor for what we do. So the study began in 1976 under the leadership of Frank Spicer. I'm the current principal investigator, and my colleague Francine Grodstein is the director of the study. 121,700 U.S. female registered nurses answered the baseline questionnaire. They were 30 to 55 years of age at that time, and we've been following them every 2 years continuously. As I mentioned, the initial major hypothesis was oral contraceptives and their potential impact on risk of breast cancer. We were also interested in smoking and cancer in women because data was sparse, hair dyes, postmenopausal hormones, and a few other risk factors for cancer. The way the study operates is that every 2 years we send a follow-up questionnaire to the women. So we started that follow-up cycle in 1978, and that's continued up to the present. This questionnaire enables us to update exposure information about all of the material that we ask the nurses. So reproductive factors, body weight, smoking, hormones, other medications, diet, physical activity, quality of life, and many other factors that we can ask about in a questionnaire. As new exposures come up, we evaluate them and add them to the questionnaire, replacing other items. We don't want to overtax these nurses, so space on the questionnaire is at a premium, and we have big battles in our meetings over every square inch of the questionnaire to make sure that we're getting the maximum scientific information. So this is just a schematic outline of how the study's been going every 2 years, questionnaires all the way up into the present. We have over 90% follow-up 36 years later of the living nurses. Many have died, but of the ones who are alive, over 90% complete the follow-up questionnaires. This is an incredibly loyal, dedicated group, and the high rate of follow-up is the main strength that enables us to have valid results. Because in a prospective follow-up study, if you don't get good follow-up, you don't know if something different is happening to the women that you don't follow. And if that follow-up is differential with respect to the exposure information, then you can get invalid results. By having a high degree of follow-up, we ensure valid findings. Well, the study was working out great in the 1980s. We realized that we needed another cohort of younger nurses because we weren't capturing the important exposure information, especially for breast cancer, that occurs in early life. So on the heels of the success of Nurses' Health Study 1, we founded Nurses' Health Study 2 under the leadership of Walter Willett, who remains the PI of that study. It's the exact same design, and we have diet information every 4 years, just as we have with Nurses' Health Study 1. So here's just an excerpt of the original Nurses' Health Study questionnaire, 1976. This is the old days. Computers were very rudimentary. We didn't have desktop computers at all. And you can see it was fill in the blanks. What is your date of birth? What is your height, your weight? And so on, basic demographic and basic exposure information. All of this had to be key entered, a very laborious and error-prone process. In later years, we switched over to an optical scan form. So this is an example of a recent questionnaire where instead of having the nurse fill in the information, they do little bubbles just like you do on your standardized tests. So we ask, for example, do you currently smoke cigarettes? No, yes. If yes, how many? And we have pre-specified categories instead of a fill-in. And the reason for that is that when you get fill-in information in your analysis, you always go to categories for your analysis. So we just skipped that step, went straight to categories, and this enables us to skip the step of keying in the data. We can have it directly read in. Many other advantages to optical scanning. So you, in your class, have a data set from Framingham, and like students of epidemiology all over the world, you have variables and you manipulate the variables. And few people actually step back to think that these data, they came from someplace. They didn't just float out of the air, but they actually exist only through the efforts of the participants and the staff that process the data. So what you see here is a picture of our coding room where every questionnaire is scrutinized to make sure that the bubbles are filled in correctly, and all of the open-ended questionnaires responses are coded properly. For example, brand of cigarette or brand of margarine that they use. People look up these long lists of margarines or multivitamin brands, and they get the right number, and then they bubble it in to the questionnaire. So a lot of work, human effort goes into creating these variables that just pop up on your computer screen. Well, we didn't just stop at questionnaire data. We also collect blood samples, which enables us to look at plasma, red cells, DNA, and other interesting biomarkers that circulate. Cheek cell samples to give DNA for women who didn't give us blood. We have urine samples. We get tumor blocks for the women who report that they've had cancer. We go back to get the medical records, and then we also go back to get the tumor sample so we can do analyses. We get blocks from benign breast disease as well as malignant breast cancer. We have mammograms. We collect toenails that allow us to look at trace element levels in these individuals. So I think we have the largest collection of toenails anywhere. So this just depicts the addition of biomarkers. So we started off with the simple questionnaire that I showed you. 1980, the brainchild of Walter Willett was the food frequency questionnaire, now used all over the world for dietary studies. So we added that. Then later we realized that we could collect blood through the mail. We just sent these women a kit and said, please go to your doctor or your clinic, get your blood drawn, and send it back to us in a special FedEx courier. We got a second blood sample, and we started collecting urine and also buccal samples. In Nurses Health Study 2, we did the same kind of thing, but we added a step that so far no other cohort in the world has, which is timed collection of blood and urine according to the menstrual cycle. So with premenopausal women, we had many thousands of premenopausal women. The blood levels of hormones change very dramatically during the menstrual cycle, and so to examine the influence of hormones on breast cancer risk, you really need timed samples, and we got them because the nurses are so wonderful. So first blood collection. This is just a schematic of the menstrual cycle. Follicular blood, during the follicular phase of the menstrual cycle, blood was drawn, and then second blood collected later. Finally, we sent a postcard to the nurses afterwards to find out when their next cycle began, so we could count backwards to figure out when ovulation was most likely to have occurred. This is a picture of a small part of our large freezer archive. We have many liquid nitrogen freezers to store these samples. We do nested case control studies within these blood samples, and it generates lots of new samples, new aliquots for study of lots of different things. So this is a picture of one of our research assistants dipping into our precious archive to get some blood, and here's a tech preparing the blood samples for analysis, which we send the samples to collaborating labs all over the world. We try to identify the best labs who can get the most accurate data with the least amount of sample at a reasonable price. We've had great success with many discoveries based on these blood samples. So I thought it'd be interesting just to go through a couple examples just to give you a little flavor of what the Nurses' Health Study has contributed. And it's impossible, even if I took up the entire course, let alone just one chunk, I still couldn't give you the full scope of what the studies have done. Just to give you a sense of it, if you take all of the Harvard cohorts together, the Nurses' Health Study 1, Nurses' Health Study 2, Health Professionals Follow-Up Study, which is our cohort study in men, which you'll hear about more later. If you take all those together, basically the unit publishes about 300 papers per year. So it's a huge scientific output that's had a big impact. But this is just a little sample of a couple items that might be of interest. So first, just a word about breast cancer. We know from the Nurses' Health Study, and subsequently confirmed by other studies, that postmenopausal obesity is associated with increase in risk, but the mechanisms have not been completely clear, and also it's not been clear whether weight loss after menopause would actually decrease risk. So we took a look at both of these in the Nurses' Health Study. In this analysis, we looked at the risk of breast cancer according to plasma levels of estradiol. Now, estradiol in the blood in postmenopausal women mostly comes from the adipose tissue, from fat. So fatter women have more estradiol circulating. We wanted to see whether the women with higher levels of estradiol had a different risk of breast cancer than women with low levels of estradiol. And we looked at this by examining the relation in 3 types of breast cancer. In the first, we looked at breast cancer that expresses estrogen receptor in the tissue, estrogen receptor positive breast cancer. And here we see that women with the highest level of estradiol here have over a 3-fold increase in risk of breast cancer compared to the control women here who had the lowest level of estradiol. In contrast, for the 2 other forms of breast cancer, the estrogen receptor negative and the progestin receptor negative, you can see that essentially there's no relation. The risk of breast cancer according to level of estradiol was only seen in the ER positive, PR positive breast cancer. So this lends a specificity to the relationship and strongly supports a cause and effect interpretation. The second question was, does weight loss after menopause reduce a woman's risk of breast cancer? Now, the ideal way to study that would be to do a randomized trial of weight loss in postmenopause and see what happens. So that would conceptually be a good thought, but operationally it would be next to impossible to conduct. It's very hard to do successful studies of weight loss long term. So instead we looked at this in an observational way. So we looked at women who didn't lose or gain much weight. So this is the reference category. And then we looked at women who either lost 10 or more pounds since menopause, kilograms, or gained 10 or more and various levels. And you can see a beautiful dose-response effect of weight with breast cancer risk. So compared to women who didn't lose and didn't gain, the women who gained weight had a further increase in risk, but the women who lost weight had a decrease in risk. So this points to a specific behavioral action that women can take after menopause that will very substantially reduce their risk. These women had a 40% reduction in risk compared to those whose weight remained the same. As part of our efforts for breast cancer study, we've collaborated with the National Cancer Institute, the CGEMS project, for genetic analyses of risk variants for breast cancer. And we've made several important discoveries, and perhaps the most important discovery was that these single nucleotide changes add only modestly to breast cancer risk prediction. It's really mostly in the behavior, not the genetics. Shifting gears, another example is in colon cancer risk. We found that individuals who used aspirin regularly had substantially lower risk of colorectal cancer, and this has been confirmed in other studies. We took it a step further, and we looked at the expression of COX-2 in the tumor tissue. So this is a good example of taking questionnaire data, getting a finding, and taking it one step further using the biologic resources of the cohort, in this case the tumor tissue. We found that aspirin lowers the risk of colon cancer, but specifically lowers it for tumors that express COX-2, and that there's a strong link between aspirin and COX-2. So this again provides very strong cause and effect interpretation for the aspirin findings. Aspirin use also was associated with lower risk of death from colorectal cancer, and there's a suggestion that even breast cancer could be reduced with aspirin use. Another example is in the realm of coronary heart disease, and this example is one that relates closely to the Framingham study and shows the strengths and limitations of the two kinds of approaches, which I'll point out here. So we've identified many risk factors for coronary disease, and some years ago we asked the question, well, how much of an effect could there be just by lifestyle factors on risk of heart disease? So we identified just these 5 simple risk factors, behaviors that just about anybody could achieve. Not smoking, so this includes never smokers or past smokers, not being overweight, BMI less than 25, moderate alcohol consumption, not too much, and not avoiding the teetotaling lifestyle, physical activity for 30 minutes a day, including walking, and a good diet with fiber, fish, folate, representing fruits and vegetables, low trans fat, reasonable glycemic load, and not too much saturated fat in relation to polyunsaturated fat. So these were the 5 lifestyles. And what we found was that women who were adhering to all 5 of those had a relative risk of 0.17 compared to the rest of the cohort. We estimated that 82% of the heart disease in the entire cohort would have been prevented if all the women were following this lifestyle. That's the good news part. The bad news part is that only 3%, and these are all nurses, only 3% met these 5 simple criteria. Now, where does this fit into Framingham? Well, it fits into Framingham when you think of 120,000 women and only 3% meet these criteria. If that same proportion were to hold in Framingham, there simply would not be enough statistical power to do this. So for this kind of analysis, you need very large numbers when only 3% of your population actually is meeting those criteria. So, but that's kind of a piece of good news for people who want to avoid heart disease. So the last element, just a quick overview, is our studies of cognitive function. These have been led by my colleague Francine Grodstein. She developed a method of assessing cognitive function through a 15-minute telephone call. So we don't see our participants. They live all over the country. We can't afford to bring them in for detailed neurophysiologic testing. So we have to be creative. And Fran was very creative, developing a method which was validated by comparing it to detailed neurophysiologic testing. And she found that this 15-minute telephone interview was just about as good as a 3-hour face-to-face interview with a neuropsychologist. So armed with that validation data, then we assessed cognitive function in the cohort, and we found some important new results. First, we found that long-term regular physical activity was associated with markedly improved cognitive functioning compared to couch potatoes. Moderate alcohol, up to 1 drink per day in these women, was associated with a lower risk of cognitive decline. Another piece of good news. And then most recently, just this year, our paper came out on habitual intake of blueberries and strawberries. And these fruits in particular were associated with a marked reduction in cognitive decline. So we're starting to pick up some behavioral factors that could have a big impact on long-term health. Finally, I just want to close by acknowledging my principal investigator predecessors for the nurses study. Frank Spicer is the founding principal investigator and still remains quite active in the study. The next PI was Graham Kolditz, a longtime colleague, and he was succeeded by Sue Hankinson, who was PI for several years before I started. And here's a picture of Walter Willett, who is the founding principal investigator and continuing PI of Nurses Health Study 2. Thank you.